In this video, we will be discussing organelle function and apoptosis. The organelles we will be discussing include the smooth endoplasmic reticulum, the rough endoplasmic reticulum, the Golgi apparatus, the mitochondria, nucleus, lysosome, peroxisome, vacuoles, microtubules, microfilaments, and intermediate filaments. As a general overview, apoptosis is the death of cells that occurs as a normal and controlled part of an organism's growth or development. In our demonstrational cell, the plastic bag represents the membrane and the water represents the cytosol. The first organelle we will discuss is the nucleus. This is what stores the cell's DNA, and it is what coordinates the cell's activities, which include growth, metabolism, protein synthesis, and cell division. The next organelle is a little difficult to see in this demonstration, though it is the green and white puffball. The mitochondria is what produces the ATP and regulates cellular metabolism. The next organelle is the Golgi apparatus, which here is demonstrated by the black and gray pipe cleaner. The Golgi apparatus is responsible for processing and bundling macromolecules like proteins and lipids as they are synthesized. The endoplasmic reticulum is demonstrated by the gold pipe cleaner. The black dots on this gold pipe cleaner are the ribosomes. The rough endoplasmic reticulum, which is the one with the ribosomes, plays a central role in the synthesis and exportation of protein and glycoproteins. The smooth endoplasmic reticulum makes hormones and lipids and distributes them. It also regulates and releases calcium ions and processes toxins. The sparkly blue puffballs in this demonstration represent the lysosomes. The role of the lysosome is to digest excess or worn-out organelles, food bacteria, or engulfed viruses or bacteria. The small red balls demonstrate peroxisomes. Their function is to break down fatty acids. They also convert hydrogen into H2O2, hydrogen peroxide, and then into water. The yellow puffballs represent vacuoles. Their function is to isolate harmful materials, store waste, store water in plant cells, and balance the pH. Our last topic includes the three main components of the cytoskeleton. These are demonstrated by the very long red and pink pipe cleaners. These three components include the microtubules, which move vesicles, granules, and organelles and provide structure and motility, the intermediate filaments, which supports the cell and cell motility, and the microfilaments, which support structure. Next, we will demonstrate apoptosis. It is important to note that there are two main routes by which cells can activate caspases and enter apoptosis. However, here we will demonstrate death signals, where a ligand on the surface of a cytotoxic T lymphocyte will bind to a receptor on our cell. After the binding of the ligand to the receptor, the cell's DNA segregates near the periphery of the nucleus. The volume of the cytoplasm decreases, small blobs are produced, the nucleus and organelles fragment, DNA is cleaved by DNase, and finally the cell dismantles into small pieces. Those small pieces are then engulfed by macrophages. It is to be noted that in our demonstration, apoptosis appears very disorderly. However, in reality, apoptosis is very orderly and highly regulated.